will move from a medical physics to radiobiology. But if it hadn't been for radiobiology, that is response of cancers and normal tissues to radiation, there would not be any medical physics in the field of radiation therapy. And really, for the best outcome of radiation for cancer therapy, we have to understand how our bodies, how cancer respond to radiation. It is a great pleasure for me to invite two distinguished speakers to this webinar. Professor Iuliana Toma-Dasu, who is a professor in medical radiation physics and the head of medical radiation physics at Stockholm University, affiliated to the Department of Oncology and Pathology at Karolinska Institute in Stockholm, Sweden, and Professor Loredana Marcu, who is a professor of medical physics at the University of Oradea, Romania, and adjunct professor at the School of Health Sciences, University of South Australia. Today, they will be talking about biomarkers. Biomarkers for hypoxia and angiogenesis, as well as biomarkers for stem cells and proliferation. And they will cover the both aspects: imaging, therapy, and treatment outcome prediction. And without wasting much time, I will hand over to Iuliana. However, can I please ask you that you mute your microphones? Please also, where possible, close your video streaming so that you don't use too much of the bandwidth, so that we have uninterrupted presentation. Lastly, we will have the question time after the two presentations. Please type your questions throughout the talks into the chat. I will be monitoring the chat, and then I will ask your questions in the last 10 minutes of tonight's webinar. So without much more talking, I'll hand over to you, to our distinguished speaker, Professor Iuliana Toma-Dasu. Over to you, Iuliana.
Many thanks, Eva.
I'll stop sharing and allow you to share.
Right. Do you see my screen?
Yes, yes, perfect.
Perfect. Thank you very much for your invitation. It is a real pleasure to give this talk today, so I'm really glad to give this presentation as part of the webinars organized by IOMP. And it is not only because I find the topic very interesting, but also because it gives me the opportunity to highlight the complexity of the role of the medical physicist working in radiotherapy nowadays, as Eva was mentioning in the introduction to our talks. The times when the medical physicists were only in charge with the calibration of the beams, with the dosimetry, the planning or the delivery of the treatment, and of course the quality control and quality assurance, I would say that are long time gone when we talk about modern radiotherapy. When it comes to modern radiotherapy, the medical physicist is expected to cross the bridge, to cross a nice and challenging bridge, I would say, towards biology, towards understanding the tumor biology to a level that would allow interpreting and modeling the role and also the influence of the biomarkers on the treatment outcome, as well as the development of approaches and techniques for integrating functional imaging based on imaging biomarkers into radiotherapy. So perhaps a good start for my talk today on biomarkers for tumor hypoxia and angiogenesis would be to explain why are we interested in imaging tumor hypoxia. And in order to do that, one has to recall the basis of radiobiology, namely the fact that the main target of radiation With respect to cell killing, it's the DNA molecule in the nucleus of the cell, and that radiation interacts with the DNA through a direct mode of action and an indirect mode of action. In the direct mode of action, the energy deposited by radiation comes from the charged particles released by radiation, and in the indirect mode of action, this process is mediated by the hydroxy radicals formed mostly through the radiolysis of water molecules in the cellular microenvironment. So the energy that is deposited by radiation in this way might induce damages to the DNA strands. And if these damages are on one strand of the DNA, we call them single strand damages, and they are regarded as easily repaired by the DNA molecule because the DNA could use the opposite strand as a template to perform this repair. If the DNA breaks are on opposite strands but rather well separated, they can also be regarded as single strand breaks, and they can be handled separately with respect to repair. But if they are on opposite sides, rather close to each other, then they are a little bit more problematic to repair. So in this context, when talking about DNA strand breaks, one can introduce the so-called oxygen effect, referring to the fact that the DNA damages could be wrongly repaired, fixed wrongly, if the molecular oxygen happens to be in the vicinity of the damaged site. In this way, the damages are made permanent because they cannot be handled by the cells through the regular mechanisms of repair, and hence the cells that have oxygen will appear as more sensitive to radiation. So extrapolating, one could expect that well-oxygenated cells in the normal tissue would be more sensitive to radiation, and tumor cells that are lacking oxygen will consequently be more resistant to radiation. A straightforward question would therefore be how much oxygen is needed in order to observe this oxygen effect. Well, not much, as it turns. It has been shown in many clonogenic cell survival experiments performed in vitro, as illustrated in this cell of cell survival curves. For cell populations irradiated in oxic or in anoxic conditions, as well as in at different levels of oxygen partial pressure. So it has been shown that an oxygen partial pressure of only 2.5 millimeters mercury, for instance, will make the cell survival bend enough to see a clear difference in survival in comparison to the case when the cells will be irradiated in absence of oxygen. So this is in vitro. Based on these observations, the oxygen effect could be perhaps even better illustrated by plotting the relative radiosensitivity of the cells as function of the oxygen partial pressure in the cells. And one could observe on a plot like this that in presence of oxygen, the cells are about 3 times more sensitive to radiation than in absence of oxygen. And this steep increase in radiosensitivity with oxygen partial pressure in the cell microenvironment could even be better seen if we plot this oxygen partial pressure on a logarithmic scale, and one could set, based on a plot like this, a threshold for what we call radiobiologically relevant hypoxia, and an arbitrary threshold could be set around 10 millimeters mercury. But as I said, these are simple experiments in vitro, indicating the role of oxygen for radiosensitization. And the question is, how important are these observations for the treatment of tumors? And the answer could be found among the 6 hallmarks of cancer that tell us that cancer cells, in addition to being resistant to apoptosis, presenting a sustained proliferative signaling in absence of stimuli from outside, being able to avoid growth suppression, activating the invasion of the nearby healthy tissue and the metastatic potential, and enabling telomerase, so virtually becoming immortal in that sense, are also capable to send angiogenetic signals to form blood vessels and grow self-sustained tumors from the vasculature point of view. However, the vasculature of the tumor is far from being well organized, and that will result in an improper oxygenation of the tumor cells and the development of hypoxia in tumors. So in tumors, generally tumors are likely to present both so-called chronic hypoxic cells and acutely hypoxic cells. The chronic hypoxic cells, the chronically hypoxic cells, are the cells that are far away from the blood vessels, but not yet necrotic. They have both reduced oxygen and nutrient supply, and they are expected to be more resistant than the well-oxygenated cells, but not as radioresistant as acutely hypoxic cells, because they have reduced DNA capacity, repair capacity, and they are the cells that might reoxygenate through the so-called slow reoxygenation process that takes place during the radiotherapy course. And as I said, tumor cells could be in an acutely hypoxic state, and we refer to acute hypoxia when we talk about the cells that are close to a blood vessel that is temporarily occluded, and they are lacking oxygen but not nutrients, and hence they have a normal DNA repair capacity, and they are more resistant than chronically hypoxic cells, and they are likely to restore their oxygenation status through fast reoxygenation. They are, however, very prone to have a high metastatic potential. So the fact that tumor hypoxia is acting as an adverse factor with respect to the success of radiotherapy has been known since long time ago, and plots like this Kaplan-Meier plots in this slide, showing the poor prognosis for, as in this example, cervical cancer patients presenting either primary tumors or lymphatic nodes that lack oxygen, have been reported based on polarographic measurements of tumor hypoxia using Eppendorf electrodes in many studies and for many tumor types along the years. Nowadays, we know that hypoxia is a problem because altered expression in genes because of proteomic or genomic changes or clonal selection could lead to slowing the proliferation and cell cycle arrest, and in the G0 phase, that will further lead to increased radioresistance to both radiotherapy and chemotherapy, and hence poor therapeutic response, but also increased aggressiveness and metastatic potential, loss of differentiation, loss of apoptosis, and chaotic angiogenesis that eventually will not only lead to poor response to treatment, but also to poor long-time prognosis because of local regional invasion and formation of metastasis. So no matter how we look at hypoxia is bad news. Consequently, several biological tumor markers have been identified and investigated in relation to the treatment outcome, in particular in relation to local control. Among these biological markers, one could mention the hypoxia-inducible factor 1 alpha, the carbonic anhydrase 9, the glucose transporter 1, the vascular endothelial growth factor, osteopontin, and interleukin 8 as major markers with a role in outcome predictions, often discussed in the literature. So Among them, hypoxia-inducible factors appear to be of particular importance, since changes that depend on hypoxia-inducible factors trigger metabolic adaptation, improved systemic oxygen supply, and eventually cell survival and cell proliferation. And also changes independent on hypoxia-inducible factors promote radioresistance, resistance to apoptosis, and suppression of anti-cancer immune response. And HIF-1 alpha, these hypoxia-inducible factors, and HIF-1 alpha in particular, are acting in synergy with other factors like this glucose transporter, GLUT1, which, when it's upregulated, aids in transporting the glucose into the cells and helping thus in the cell metabolism, or the vascular endothelial growth factor that plays an important role in apoptosis. Now, the role of hypoxia markers in relation to the outcome after primary radiotherapy has been investigated for several tumor types. An illustrative example for laryngeal cancer patients, it's shown in this table. And one could see that two out of five, in 2 out of 5 studies, the HIF-1 alpha expression has been positively associated with the treatment outcome, expressed as either residual tumor presence of the residual tumor less than half a year after the treatment, or as local recurrence after more than 6 months post-treatment, or generally as time of local recurrence. The situation it's almost the same for CA9, where we have 2 out of 6 studies showing this positive predictor role, while for the other markers, no relation has been observed. So, using biomarkers for outcome predictions comes at a price. And I'm not referring here at the actual cost of the tests, but to the fact that the techniques for assessing them, the presence of biomarkers, are invasive for the patients. And hence, one started to consider how to move from invasive biomarkers to non-invasive techniques or minimally invasive techniques like functional imaging. So functional imaging of tumor biomarkers could be done, could be performed with PET, but also with MR. And I will mostly focus on PET in my talk. But if you would like to learn more about MR imaging techniques for biomarkers, I could recommend a review written by my friend and co-presenter today, Loredana Marcu, and our kind moderator, Eva Bezak, and their colleague, Paul Reed. So coming back now to PET, there are several tracers specially designed to image hypoxia. Most of them are coming from the nitromidazole family, such as F-MISO, FETA, FASA, EF5, AGX4, and so forth. And there are some other tracers like CAPR-8SM that are not belonging to the same family. The most used tracer nowadays, it's still the good old reliable, to some extent, fluoromyzonidazole that has been designed, of course, in such a way that cellular retention of F-MISO depends on the oxygen concentration in the cells, and under hypoxic conditions, this rate of uptake of F-MISO, the binding, can be much higher than under normoxic conditions. So there are quite a good number of studies assessing the oxygenation of tumors based on F-MISO. But if we look now at various tracers, all of them designed for imaging hypoxia, we could see that various tracers provide different levels of uptake and hence different discrimination between the hypoxic levels of the cells, as illustrated in this example, for copper ATSM and for F-MISO uptake as a function of the oxygen partial pressure in the same cell line. Furthermore, the uptake of a particular tracer appears to be influenced by the metabolic characteristics of the cells. Here we have the uptake of the same tracer, theta, into different cell lines. So generally, it could be concluded that various PET tracers, all designed for imaging hypoxia, could lead to different images depending on their uptake characteristics. So if we look now for the correlations between PET, or to some extent SPECT, tracers and the hypoxic tumor markers, or generally parameters, one could expect that the correlations are rather strong, while in fact they are not that strong. They are rather weak in most of the cases, are shown in this table for various markers. Some of them I already mentioned, and various PET tracers. So perhaps one of the reasons for the weak correlations that were found could come from the fact that there are no standardized protocols for interpreting and for quantifying the information in a PET image that would present in the most typical way like this, or a clear answer to the question regarding the parameter, or rather the parameters that should be selected for seeking correlations with the outcome in order to make this kind of predictions. So regarding the first question, how do we interpret and quantify the information in a PET image? There could be several answers. The most common one, the most common method for quantifying the information in the PET image, beyond quantifying the standard uptake value, of course, it's to use a reference region and to set a threshold for hypoxia based on a ratio between the uptake in the tumor and the uptake in the blood, for instance, or the uptake in a muscle, knowing that blood and muscles or normal tissue are expected to be well oxygenated and hence present a low uptake of a hypoxic tracer. Another way of handling this problem could be to use a conversion function from the relative tracer uptake to actual oxygen partial pressure of in the cells. So an example of a study aiming at quantifying or quantitatively analyzing rather images of tumor hypoxia that was performed in our group, it's shown here. So lung tumors were imaged with HX4, a novel and assumed more promising tracer for tumor hypoxia in comparison to F-MISO. And there were three modalities used for determining quantitatively the presence and the extent of tumor hypoxia. Two of them were based on this ratio between the uptake of the tracer in the tumor and in two reference regions, the aortic arch being one of them, and a muscle, respectively, and one based on a function that we introduced for converting this uptake of a tracer into an oxygen partial pressure. The hypoxic subvolumes delineated in one case, in this case used as an example, shown here, present the hypoxic target volume based on either this aortic arch as reference region in orange, or the muscle as a reference region in green, and the hypoxic target volume obtained by using this conversion function of the uptake of HX4 to PO2, a function that is also shown in this slide, together with a corresponding one for F-MISO, to point out that there are differences between these conversion functions. They have the same shape, but not exactly the same parameters that describe them. So as one could see, the volumes delineated using different methods are far from being the same. This might therefore impact on the predictions of the outcome if one would consider making using one or another of these volumes obtained using these different methods. So here we have another example from a subsequent study in which we highlighted the importance of doing this segmentation of the hypoxic target based on the relative uptake of the tracer converted to oxygen partial pressure, because it would allow us to determine not only the extent of the hypoxic volume, but also the severity of hypoxia, and hence it could be used for tailoring the treatment for individual patients. In this particular case, we have the uptake of the tracer expressed as a standard uptake value in this left panel, the map of hypoxia in millimeters mercury, so partial oxygen pressure in the middle panel, and the corresponding contour here for the hypoxic target volume based on the quantitative image of hypoxia in the middle panel, using a threshold of 10 millimeters mercury to delineate this hypoxic target volume, presented together with the gross target volume and the clinical, the gross tumor volume and the clinical target volume in this right panel. So what was said so far was in a sense restricted to looking at one single image, but tumor hypoxia is far from being a static feature. So a legitimate question regarding the parameters that should be investigated in relation to the treatment outcome predictions could be the following one. Which parameters do we select for correlating with the outcome in order to make predictions? And also, at what time point are we looking for these parameters? So here are also some examples from the analysis we've performed. So let us say that we have one patient, as the one shown here, that has a hypoxic tumor, primary hypoxic tumor, and a hypoxic lymph node, and they are imaged before the start of the treatment and 2 times during the course of the treatment. And here we have another patient presenting a hypoxic lymph node before and during the treatment. And the question is, what image and what parameters should be chosen for seeking correlations with the treatment outcome, given these images of tumor hypoxia? All of them are images of tumor hypoxia. Here are also examples from the same study of one patient that presented local control and one that presented local recurrence. And their corresponding hypoxia images assessed with F-MISO before the start of the treatment and during the 2nd and the 5th week of the treatment respectively. And here are patients imaged with F-MISO head and neck patients. And several parameters derived from these images could have had predictive value and be investigated. But the results of this study that we performed on 30 head and neck cancer patients showed that the most predictive parameter was the % decrease in the hypoxic volume segmented based on the conversion of the tracer to oxygen partial pressure from the pre-treatment image to the one taken during the 2nd week of radiotherapy. And this parameter had a sensitivity of 60% and a specificity of 91% with respect to local recurrence. This was just one example, but a recent review showed that in case of longitudinal studies monitoring the tumor hypoxia with PET, there are indicators that the pre-treatment hypoxia has predictive value for treatment failure, the same for residual hypoxia for local recurrence, and an increase in the hypoxic volume during the treatment has also positive value for predicting the local recurrence. However, there are no conclusive studies showing that the decrease in size of the tumor hypoxia during the treatment correlates with local control. So in spite of the many studies available in the literature, at the present state, it is difficult to formulate a strong statement or general strong statements valid for many cases of tumors, or at least for particular tumor types, classifications, and staging regarding the predictive values of imaging biomarkers for tumor hypoxia. I would have loved to come with a clear and strong conclusion for my talk today. So what I could say instead is that tumor hypoxia detected with PET during radiotherapy might indicate poor outcome, and that the disappearance of the tumor hypoxia during radiotherapy does not automatically lead to treatment success. And assessment of hypoxia dynamics with repeated hypoxia PET scans during radiotherapy has a high potential for outcome predictions in comparison to single assessment of hypoxia before the start of radiotherapy. Furthermore, what I would like to highlight is that hypoxia should be integrated within radiotherapy, not only as a predictive assay, but mostly for personalized treatment approaches designed to counteract the radiation resistance associated with oxygen deprivation. So, as few final considerations, I could say that imaging and quantifying tumor hypoxia does hold promises, not only for outcome predictions, but also for monitoring the response to the treatment and adapting the treatment accordingly. Standardized imaging protocols, however, and methods for quantifying hypoxia are still needed, are not yet available. If they will become available, it would be much easier to design large clinical trials focusing on tumor hypoxia assessment with functional imaging that are also very much needed for getting conclusive results. And finally, if this kind of studies will become available, radiomics approaches might also be possible, and they might shed light on some aspects that are not even yet foreseen. So that was my talk today. Thank you for your attention. And my final words on this topic would be that although tumor hypoxia is a key parameter with respect to the treatment outcome, it's not the only piece of the puzzle, and it should be integrated into the overall picture together with other parameters and factors such as cell proliferation and cancer stem cells known to influence on the treatment outcome as well. So thank you again for your attention.
Yeah, wonderful. Thank you very much, Iuliana. I will jump immediately to Loredana. Loredana, could you please be so kind and share your screen?
Yes.
Thank you. I am recording the questions in the chat, and as I mentioned, we will have a Q&A session after the presentations. Loredana, over to you.
Thank you very much, Eva, for your kind introduction and for inviting me to deliver this presentation. And thank you, Iuliana, for paving the road for my presentation, as the two talks are obviously strongly linked. We are both talking about the biomarkers from imaging to outcome prediction, and particularly for a solid tumor where we don't only deal with hypoxia, but we deal with several other features that strongly influence the response to therapy. We have to see the big picture and to discuss them all. So, no doubt there are several clinical challenges when we talk about the management of solid tumors, and beside the primary tumor, where we look at the preservation of the critical organs, so the organs at risk, the side effects, the treatment of chemo, radio, immunotherapy, so on and so forth, we also have to look into the systemic therapy, because distant metastasis remains a big problem nowadays. But from a radiobiological point of view, again, we have several other challenges, and these are the ones that we are pointing out or highlighting today. So as Iuliana mentioned, tumor hypoxia, it is indeed one of the greatest challenges and one of the biggest culprits for tumor recurrence and treatment failure. But beside hypoxia, we have other features of tumors such as repopulation and radio resistance. I would call it intrinsic radio resistance, or the inborn radio resistance, because if we just mention radio resistance as such, we have to ask, why is the tumor radio resistant? Because it is hypoxic, because it has a high proliferative ability, because of the large fraction of cancer stem cells, so on and so forth. So I presume we have to make the difference between intrinsic radio resistance and just general tumor resistance to treatment. And again, the management of disseminated disease, which I will highlight a bit towards the end of my presentation. I'll introduce the so-called circulating tumor cells, which are under research these days, because they present a very important aspect when we talk about outcome prediction. So as I said initially, there is no doubt that over the last few decades, radiobiology has developed a lot. And that is very good, because we know that radiobiology is the science behind radiotherapy. Due to radiobiological advances, the imaging techniques have evolved greatly, as you have seen from Iuliana's presentation as well, which helped us in radiotherapy to improve tumor targeting. And because most of us are physicists, we don't have to forget the physics that went hand in hand with the previous developments. And we now have a much better matching between beam quality and tumor properties, reason why several centers now treat with protons or heavy ions instead of just photons and electrons. Still, there are several challenges on the front of treatment resistance, tumor recurrence, distant metastasis. So everything has evolved, has developed over the last decades, but we are still facing several problems here. Why is that? Well, I would say because we don't really see the forest for the trees. Especially as physicists, as Iuliana very nicely said in her introduction, we were used to deal with the technical aspects, with dosimetry, with quality assurance, with treatment planning, but we shouldn't forget radiobiology, that actually is the basic, the fundamental basis of radiotherapy. So what we don't see are exactly these issues that were pointed out before, and that I'm going to point out during my presentation, that hopefully now you can see from my screen. So it's the hypoxia that has been discussed, is the intrinsic radio resistance, the growth kinetics that are linked to the proliferative ability of tumors, the circulating tumor cells, and the cancer stem cells, which are becoming, I would say, a big headache, since we know their properties and we know what they are able actually to do. 40 years ago, two big professors and researchers, Lester Peters and Rodney Withers, said that the clinical problem of tumor radioresistance is largely the inability to predictively identify its cause in the individual patient. Well, 40 years later, probably we know a bit more. And we definitely know that tumor microenvironment is patient-specific, which requires personalized treatment. We cannot put patients under the same hat and treat them all the same, because then we'll get different effects. And this would lead to the requirement to identify biomarkers to predict the response to therapy. So the question is, again, what makes a good biomarker? We have seen several presented in the previous talk. We'll see a few more in my talk, but there are a few requirements that each and every biomarker should meet in order to classify as an adequate one. Biomarkers are objective, and quantifiable, or should be quantifiable, characteristics of biological processes. Ideally, a biomarker should be highly specific to that tumor type or that particular feature of the tumor, specific to hypoxia or proliferation specific or cancer stem cell specific, and so on and so forth. Ideally, again, it should be highly sensitive, so we know that we can trust that biomarker. It should have a predictive power, and again, for the patient's comfort, it should be non-invasive. Now, if a biomarker meets all these requirements, there are several applications for biomarkers, from diagnostic to risk assessment, but probably one of the most important ones now is patient stratification. We can hear throughout the lectures and books and articles about personalized treatment in radiotherapy, which is really the focus of not just medical physicists, but biologists and oncologists, and it should be the focus for the future oncology, because we are not progressing in the way that we would like, or we or others might have predicted like 50 years ago. So patient stratification is an important factor in this sense. Now, just a small bracket regarding the market of biomarkers. If you look at this chart, you would see that the US market predicts a huge investment by 2025 regarding research in cancer biomarkers. So we are talking about nearly 34 billion US dollars by 2025. And if you look into that chart, personalized medicine would play a pretty important role and would get some finances, hopefully, for their further development. But let's see, where do we stand now? And let's start with cancer stem cells. I'll make an introduction to this particular terminology in case some of you are not very familiar to cancer stem cells. So first of all, these cancer stem cells, or I'll call them for short CSCs, are a subpopulation of cancer cells that originate from normal stem cells. So they have this stemness in them inherited from their healthy counterparts. Now, usually tumors have small amounts of cancer stem cells, even less than 1%. And you might think that, oh, this is negligible, but you are wrong if you think that. Even that very small percent of cancer stem cells, they have very powerful properties, as you can see them from this table, because they can live for a very long time during several divisions, and actually many researchers label them as immortal because they proliferate indefinitely. Another important property of cancer stem cells are the fact that they can recreate themselves by symmetric division. What does this mean? It means that in mitosis, when the cell divides, actually a cancer stem cell can divide into two cancer stem cells. So you imagine from here an exponential growth of these cancer stem cells that can actually proliferate the tumor, develop the tumor, but even regrow the tumor during treatment, or after, even worse, after treatment, what we call recurrence. They also preferentially reside in certain microenvironmental niches within the tumor, which could be like a hypoxic region. So all these properties make them fairly strong cells as compared to other cancer cells that don't have this stemness in them. We are aware of the clonal evolution. Even if you don't have like a strong radiobiology background, you might all be aware of the initial or older theories that would say that cancer starts from one cell that has undergone several mutations. That's the clonal evolution. a side of this clonal evolution is the classical CSC model that says that actually a tumor starts from one cancer stem cell that further divides into other cells which can be non-cancer stem cells or differentiated cells that don't have the stem-like properties. However, today we know the truth. And well, the ugly truth, because the new model says that we are dealing here actually with a tumor cell plasticity. What does that plasticity mean? It means that a cancer stem cell, once it divides into a non-cancer stem cell, actually through certain biological processes, it can regain that stem-like properties. So we are talking about a bidirectional conversion from stem to non-stem and vice versa, which means that these cancer stem cells, again, have really powerful properties, so they must be monitored and targeted. Why is that? Well, from this very simple chart that has been published in Nature like 20 years ago, you can see what a cancer stem cell can do. And I was saying this at the beginning of my presentation, that one single cancer stem cell can grow or can regrow actually the tumor. As you see it from this chart, if one starts with a heterogeneous tumor that also has cancer stem cells, and you target it with either drug or why not radiotherapy, and you kill all cells but cancer stem cells, that tumor will actually shrink because you got rid of a large number of cells that are non-stem, but that single cancer stem cell actually can regrow the tumor. However, if from the beginning you target cancer stem cells, you are left with the non-stem cancer cells that don't have such powerful properties. They can't proliferate indefinitely. Therefore, eventually the tumor will degenerate. So what's the conclusion from here is that we really have to target cancer stem cells in order to eradicate them. So here we are talking about biomarkers and the CSC markers for imaging and targeting. Now, so far, there are three CSC marker categories: cell surface markers, internal markers, and functional markers. But out of these, the first one, the cell surface markers, have developed greatly. So greatly that actually I tried to find a table that I could fit in my presentation in one slide, but I wasn't able because of the very large number of surface markers for cancer stem cells that are already available. I'm just naming a few here, such as the aldehyde dehydrogenase, and lots of cluster of differentiation. You see the numbers next to them. Why are they so many? Because research shows that certain markers for stem cells are specific to certain tumors. So for example, you use a group of cancer stem cell markers for lung tumors, but you use a different one for head and neck, or for glioblastoma, or for breast cancer, and so on and so forth. Now, most of these studies have been undertaken in vitro or in vivo in animals. That's why, unlike the previous presentation of Iuliana on hypoxia, where actually there were several human studies that could lead to a final endpoint or a conclusion on the predictive power or prognostic power of hypoxia, we cannot say the same thing about stem cells, because the research is much younger on these, and the markers, as I said, they are still in a preliminary phase, being mostly used in animal studies. And you can see this actually from this table. What I'm showing here, it's also a confirmation of cancer stem cells which were identified through immunohistochemical staining. confirmed by PET tracers. You recognize some of them that were previously discussed. Copper ATSM, it is fairly fashionable nowadays. It has very interesting properties that actually show specificity towards hypoxia, proliferation, and also cancer stem cells. So if using copper ATSM, one would see a PET uptake of this radio tracer. Definitely we are dealing with a fairly aggressive tumor because we have something in there. But also iodine 124 and 125 has been trialed as a PET tracer and correlated with immunohistochemical staining through different markers. You see the CD markers, these are the cancer stem cell markers, showing that actually they have specificity towards cancer stem cells. But again, what I just mentioned before, all of these were in vitro animal studies, or in vivo in xenografts. So colorectal brain tumors or lung cancers, they all showed considerable percentages of cancer stem cells, reason why they have been heavily studied. Next, a few words about tumor proliferation. Probably you are more familiar with this aspect of cancer growth than with the cancer stem cells. And we all know that cellular proliferation is a prerequisite for growth and development, even in normal tissue. However, one of the hallmarks of malignant growth is the uncontrolled proliferation, which is definitely something typical for cancer cells. That's why there are several parameters in radiobiology that have been linked to tumor proliferation, such as the labeling index, the mitotic index, the potentially doubling time, the volume doubling time, or the kickoff time for rapidly proliferating cells. All these proliferation kinetics actually help us to differentiate slowly proliferating from rapidly proliferating tumors, which is a very important aspect when designing radiotherapy, because this proliferation kinetics would dictate the type of treatment required. For instance, for rapidly proliferating tumors such as head and neck cancer, hyperfractionated radiotherapy is much more effective than the conventionally fractionated 2 gray a day, while for slowly proliferating tumors such as prostate, hypofractionation is more effective than conventional radiotherapy. So these are all dictated by the radiobiological properties. Furthermore, a number of pathways and genes are involved in tumor proliferation, and these are being used for targeting. Two I have pointed out here, which are probably the most known ones and the most important ones, the EGFR, endothelial growth factor receptor, and the KY67, which is a nuclear protein that both of them mark actually or highlight the proliferative ability of tumors. So this EGFR, as I said, it's one of the most commonly altered genes in cancer through mutations, overexpressions, or amplifications. EGFR signaling drives several important cellular responses, regulates cell proliferation and apoptosis. Therefore, the inhibition of the EGFR activity suppresses these pathways that control cell growth. What is the conclusion from here? That we should target it. We should find anti-EGFR targeting ways. Tumor repopulation also is an important aspect during radiotherapy, because certain tumors have the ability to repopulate much faster than before the treatment, and this is what we call accelerated repopulation. And here we are back to cancer stem cells, because several mechanisms behind accelerated repopulation have been identified, but probably the most important one that was show to be is the loss of asymmetrical division, meaning the gain of symmetrical division of stem cells that can actually repopulate very fast the tumor. Now, to link the two properties together, I'm showing here two fairly recent papers, one on lung and one on head and neck cancer, saying that actually the EGFR mutation can activate and can stimulate cancer stem cells, and which was showed by labeling of cancer stem cells in a population of cells that show EGFR mutation, and they presented high aldehyde activity and elevated CSC marker expression. The other study, the head and neck, said a similar thing, that EGFR has a possible role to regulate the stemness in head and neck cancers, meaning that EGFR has to be inhibited in order to decrease the proportion of cancer stem cells. I won't go into too many details. This is a table similar to what you have seen before for the other properties, pointing out the fact that positron emission tomography, SPECT, and magnetic resonance imaging, they all have the potential actually to image, to use imaging markers that would be specific for proliferation. And one of these is actually the FLT, which shows a decrease, shows a very nice monitoring of the lung tumor during treatment. We don't have that much time, so with circulating tumor cells, I'll only say that these are critical for treatment monitoring, being those cells that actually leave the primary, get into the bloodstream, and then cause metastatic sites. And several of these cells present themselves as cancer stem cells. And since we know already the properties of cancer stem cells, they can start those distant metastatic sites that create further problems in cancer therapy. And that's my the last paper that I would like to share with you, a research done by an Australian group showing that during radiotherapy there is possible to mobilize tumor cells that would circulate actually in during radiotherapy and would cause those micrometastases, meaning that circulating tumor cells are by themselves prognostic biomarkers. So, in summary, there are several available biomarkers. However, there is no gold standard yet, and probably there will be no gold standard due to the variety of features that influence tumor control. Perhaps a combination of biomarkers will be a solution for the future, but definitely more work is needed in this sense. And people started to work on radiomics that actually is a bridge between medical imaging and personalized medicine, because you can go into details that are not seen by the naked eye, and you might draw a better conclusion in order to personalize treatment from there.
Wonderful. Thank you very much, Loredana. I better proceed with the questions right away, if that's okay. I'll welcome Iuliana back to the topic as well. And I'll start with Iuliana. Iuliana, hypoxia is major microenvironment of tumor governing tumor control. Does the tumor control, is it dependent on hemoglobin level of a patient? If you have anemic patient, would you expect higher levels of hypoxia?
Well, the oxygenation of the tumor does depend on the oxygen content in the blood vessels. And in that respect, as a straightforward physical process of oxygen diffusion from the blood vessels towards the cells, it should be a difference. However, I am not sure if there are conclusive clinical studies pointing out that hypoxia could be much more severe in this kind of patient.
Yeah. Okay. Loredana, do these predictive biomarkers differ in conventional therapy or particle or carbon therapy?
No, they are not therapy related. They are actually tumor property related. So that's here we are talking about radiobiology, not radiotherapy. So once you identified the radiobiological problems, you can actually personalize the treatment by using the best option for that patient. And you might say that proton therapy might work better for that particular patient because of, let's say, the high proliferative ability than conventional therapy.
So therapy is more of a decision-making process after you know the other characteristics of the tumor. Yeah. Iuliana, which modality of imaging is the ideal for tumor hypoxia imaging, PET or functional MRI?
I wouldn't say that any of them is ideal. I would say that they complement each other, and this opens up for a rather lengthy discussion, I would say. If we think about resolution, then obviously MR will be a better choice. If we think about the way of interpreting the image, there is a more straightforward relationship between the uptake of a hypoxic tracer in PET than about the surrogate markers in MR. If there is about the invasiveness and the fact that we do inject a radioactive tracer in the patient and the radiation protection point of view, then MR will be the choice and not PET, and so forth. If we think about MR LINAC and the opportunity to image multiple times during the course of the treatment, again, MR will be the choice. But I would not tilt the scale in any way at this particular moment, I would say.
One subsection to you. How dependent is the normalized uptake metrics on the delineation protocol?
I would say that it's the other way around. Depending on the normalization of the uptake of the tracer, the contour will be different.
Yes.
So, yeah.
And there is some subjectivity there?
Well, there is the intrinsic subjectivity because of the poor resolution in the PET image, yes.
Yeah.
Yeah.
Loredana, is there does the number of stem cells depend on the type of cancer, or is it related to the tumor volume as well?
Yes, it's interesting. I don't know, it might work both ways, though there are studies that show very different results, even among the same histopathological type of cancer. For head and neck, there were reports showing below 1%, there were others showing 13%, which is a huge difference, you can imagine. So I don't think there is a yes or no answer to this question. As I said, it is not just tumor type, but also patient dependent as well. It's much more complex than this.
Iuliana, is VEGF biomarker for prediction of complication?
In the normal tissue, I'm not aware of that.
Not aware of that, yeah. Loredana, is the CSC, if the CSCs are bidirectional, how can you just target CSCs to eradicate the tumor?
Yeah, that's a good question. It's not that they switch all the time and left and right. So it's like, I'm here, I'm not here, I'm here, I'm not here. Important is to catch them in that stage when they show stemness, because we are targeting stem cells. If they differentiate, we don't have an issue with the differentiated cells, because there are, I couldn't go into those details, but there are treatments, there are actually agents that target those cells and can deal with them. The problem is the cancer stem cell that has to be targeted when it's cancer stem cell, or target them with an agent that doesn't let them differentiate. So it doesn't messes up the big picture. So you know that you are targeting, you are actually marking and targeting the cancer stem cells.
Yes. It's really much complex than we originally thought, that you just use the radiation and eradicate the tumor. But we forget that the tumors, they have their own repair and response and protective mechanisms.
Absolutely.
So
Absolutely.
they're definitely trying to counteract the damage.
Exactly. We don't have imaging that performant with such a resolution that would allow us to see actually each cell and what's happening there. So you would say that the tumor is eradicated and you might be left with a number of cells, out of them one cancer stem cell that can actually continue the growth there.
Iuliana, in the studies that you showed, is there any concern over repeated PET imaging and increasing the patient dose? Should this be subtracted from the radiotherapy dose?
Well, it has to be regarded in some sense as the price to pay for preventing the re-irradiation of the patients in case of local recurrence, if that is
Anyway.
the case, yes. So from the radiation protection view, but also from the cost effectiveness point of view, one extra PET image or perhaps two extra PET images could be clinically and financially motivated. But one has to integrate these images into the workflow and, of course, confirm their use through clinical trials before really going for, repeated PET images,
Yes.
just for the sake of exploring what happens.
Yes. Is there a correlation between a collagen content and organization of stroma and hypoxic status? Should biomarkers related to collagen content and organization be studied together with hypoxia biomarkers?
As far as I know, there are fewer collagen fibers and more rigid, if I can call them like this. So there is a marker called COL1 in hypoxic tumors. But a one-to-one correlation study between COL1 and marker for hypoxia, I cannot recall.
Loredana, do you have any information on these rates of de-differentiation from tumor cells to cancer cells? And how does it compare to cell division rate? You know, how do you produce cancer stem cells faster, with division or de-differentiation?
Yeah, that's again a very good question. I won't give you figures because I'm not aware of any figures. I haven't searched actually the literature for that, but that's a good point to have some quantitative measures. I gather that a more powerful property would be still the repopulation through cellular division.
Especially the loss of asymmetric division, yes?
Exactly. Yeah. So there is plenty of literature pointing out this property of cancer stem cells and the fact that that indeed the symmetrical division is responsible for tumor regrowth and repopulation during treatment. So probably it's a more like quantitatively is like a higher rate than the de-differentiation.
Yeah. Iuliana, what is the current status of hypobaric chambers to overcome hypoxia?
Well, I'm not really quite sure. They were very much in fashion for a while, and then mostly, I guess, because of logistics, the technique has faded away in conjunction to radiotherapy, and also because it was not quite clear how to time exposing the patients to high oxygen content in a hyperbaric chamber and the radiotherapy treatment. So there are radiotherapy centers that are more active in promoting this technique for radiosensitization than others. But I wouldn't say that there are, well, no, that it's something that it's very probable.
I sometimes hear that the pharmaceutical companies are not interested because it's a cheap solution.
That could be as well. So I would say that hypothermia is more present on the market than hyperbaric oxygen chambers.
Due to time, I will finish with last question. Loredana, do you have any idea why there is a protection of normal tissue in flesh radiotherapy, proton or photon?
There are still several questions around it from a radiobiological perspective. Probably the oxygen depletion would be one of the main reasons that would differentiate the normal tissue from the cancer tissue, and that's why flesh would protect the cells and at the same time have nearly the same tumor control as conventional irradiation.
Okay. Loredana, may I please ask you to unshare your screen, if you could be so kind? So in conclusion, this has brought us really to the end of the IOMP webinar tonight. I would like to thank our wonderful speakers, Professor Marcu and Professor Toma-Dasu, for excellent presentations that are perhaps more enlightening and outside your main scope of work as medical physicist, but that are equally important if you want to deliver your practice in the most safe and efficient manner. And before I close, I will share my screen for one last time. I would like to remind everyone that our next seminar is on September 23 at the same time, and it will be on proton facility shielding, and it will cover regulatory and design aspects on proton shielding. There are currently about 90 proton facilities and heavy ion facilities around the world, and perhaps another 100 are being planned. So this is a really important topic where perhaps proton shielding is not covered in our medical physics curriculum. So I invite everyone to participate in the next webinar. Anyway, one more thanks to Professor Marcu and Professor Toma-Dasu. It has been wonderful spending my late evening with you, and I hope that our participants have learned lots of interesting and insightful new knowledge tonight. Thank you so much.
Thank you.
Thank you very much for your kind words and for inviting us. Thank you.
Thank you.